We would like now to start a report of the Breakout groups. We'll start with skin, going to lung, going to kidney, going to liver, and we'll start with skin. The explicit demand of Ido was that we start with a joke, which is not so much of a joke. It's a story about a very famous politician and very famous soldier in Israel who had very large body dimensions, and he used to say, usually to people who asked him, why he's so fat.
You could say Arik Sharon.
Well, I didn't, but he was often asked, how does he manage? Why doesn't he go on a diet? He said, well, that's not a problem of fat. I just have a very, very thick skin. That's why we are going to start with skin, and Muz and I discussed it yesterday a little bit, what should be discussed. I think there was a very good discussion over it, which in a way had to echo here and there some of the things that we heard last night concerning the kind of third generation periodic table. And I think one of the main challenges in the discussion was to see to what extent one can reduce complexity without losing information to a level that can justify looking at what is being done here as a kind of a periodic table of the elementary components of life. So perhaps we start with some description of the skin, and we'll just talk about it as go along, and then at the end, we'll leave a few minutes for a summary of the points that were made by close to, I think, 20 or 30 comments by the people who participated, which I think was very inspiring.
So I'll just summarize some of the challenges that we discussed, in that it is a huge organ, 1.8 meters squared of body surface area. It is also a complex organ in that it has many substructures within it and cells. And if you look at it as an organ, because of the size, it actually has many more immune cells, for example, than 5-liter volume of blood, which is the average circulatory volume. So during the breakout sessions, we sort of had general categories. One of it was actually, how do we define what is actually skin, and what will be the scope of a skin cell atlas? And I'll come to some of those points later on. Some of the general categories that we have to consider in how we actually implement and execute the atlas acquisition, and some special higher order considerations that may actually not necessarily be very obvious at the sort of cell level. And also sort of, really I think the main level is how do we define the boundaries and scope of the cell atlas. So if we look at the skin in terms of defining it, it is the outer covering layer of the human body. What are its borders? And I think this is quite difficult to know where it begins and where it ends, because it has some features: epithelium, keratinized, has stratum corneum, which is the dead skin layer. But where does it stop? You've got the vermilion border of the lip, you've got the anogenital site. One has to decide. So where do we decide what will be the first phase of skin that we're going to analyze? And then we had some general categories here. And I'll just go through some of them more quickly. And the first is actually macroscopic anatomical variation. And you can see how the skin is very different in the scalp area versus the sort of palmoplantar surface. And then you've also got the sort of dorsal surface which is UV exposed versus the one that's not UV exposed. So I think one of the big discussion points here was, how do we actually do the first stages? Do we actually focus on a small group of individuals and sample a wide variety of anatomical sites? And if so, how do we demarcate these sites? What would be the most appropriate? And where would these individual source come from? Would it be healthy living donor? Would it be a transplant or cadaveric donor? So all of these things have to be considered, because easily it becomes exponential, the number of sites that one has to analyze. And there was a very good comment from Ron Germain. Maybe skin is one structure, perhaps, which could be surveyed as from a spatial perspective to decide how then we go about defining the anatomical regions that we want to study. And then also, of course, the interface areas, which I alluded to, the vermilion border of the lip being one of them, and then some other skin-anchored tissues such as nail, and the nail bed, and do we leave them out in the first phase of the skin atlas work? And then we come to the sort of microanatomical stratification in terms of, you've got the three main layers, the epidermis, dermis, and then the hypodermis. And, how you view the skin in terms of the spatial imaging, because for high throughput, perhaps this matters. But if you want to do three-dimensional imaging, then it doesn't matter. But then that is quite time and computationally intensive. And so how do we then understand the structures within the layers? I mean, it's much more about how do we use the data to understand biology and actually capture what's important from the perspective of skin as a tissue. The epithelium has got the different layers, the basale, spinosum, corneum. How do we understand the relationship between the layers and ultimately the three-dimensional reconstruction? And sort of the functional layers, functional anatomy, I think is quite an important concept within tissue. I'm not going to go too much about all of the kind of like variabilities in terms of what one can find, but I think the main messages were how do we approach this in a sort of like achievable manner, and then other questions, which really comes down to what is actually the cell atlas going to be about? Because we've focused on cell as an entity, but in many tissues, including the skin, the extracellular material is very important. And that is what provides it the mechanical strength. So not just the rigidity of cell types, the basement membrane, the tight junctions, but also the collagen structures, which we are not evaluating, which provides the mechanical strength of the different anatomical sites of skin. So this was a question that was very much heavily discussed, in terms of going beyond the cell and outside the cell.
Yeah, perhaps just add one element that did come up. One of them, as mentioned here, is in the skin, since many of the functions are really very much dependent on the structural relationships between and mechanical properties of the different cellular components and the extracellular components, how we make sure that we don't lose them in the analysis and we look at them. The other issue, which is, I think, especially important and emphasized in the skin, is the fact that the skin is more susceptible to perturbations and unexpected perturbations than many other organs. Basically, the body, the homeostatic stable systems are making every effort and developed throughout evolution to make sure that some of the organs operate under more or less stable conditions. And when it comes to the skin, it's obviously exposed all the time to perturbations, a very large number of perturbations. The result of it can be that the problem of cell state versus cell type becomes a much more demanding and challenging issue, because many of the changes that we might find are the ones that are induced by specific environmental cue, may it be a cosmetic ointment or may it be UV and so on and so forth. And there's just an example, small example of perturbations that are there. And I think that's something that appear to be an issue that requires very serious attention. If we try to deal with the perturbations in the entirety and locations in the entirety, we get a huge number of, we'll have to make huge samples which are beyond the scope, probably, of the program. And we have to set priorities and figure out what we gain from them and what we're losing from them. Especially the issue of separating and cells into individual cells, suspending them and looking at their properties, versus keeping them together and look for the features when they are part of the system, of the tissue system in situ. And there are other parameters, perhaps you would like to continue.
Yep, so it's sort of similar to some extent with perturbation, because there is age, which is, as a temporal element which affects both the microscopic and the macroscopic considerations. And this is intrinsic, sort of from womb to tomb. And then I won't bring so much of the perturbation, but just to add that there is one thing about assessing the impact of these factors, and this is by no means a comprehensive list, but these are things that can actually affect the outcome of any study on a limited number of individuals, but also in the kind of like extension of the work going forwards. How do we then evaluate the sort of consequences of these perturbations on the skin and its composition? And then we came to sort of, technologies and actually kind of like how do we actually process the skin? And there was quite a lot of discussion on how do we process skin, and is this going to be a standardized protocol? And I guess to some extent there are many ways of doing this, and to a large extent researchers have used the protocols that best suit their research questions. So for example, studying viral infections, you use a method that preserves viral recognition receptors, and if you're pretty much studying immune cells, you'll use one protocol, or if you're studying keratinocyte, you use a different protocol. So how does the community have access to some sort of a standard way of going about it? And then how do we, what sort of technology? And at the moment, there are sort of like two main branches in terms of the transcriptome or genomics-based branch, and then there's the spatial branch. And it was very clear that the spatial branch, the technology was not quite there to sort of meet the demands. And therefore, as a community, we should try and sort of encourage the sort of development of high throughput imaging and extending it to the wider properties. And I think some of these we've touched on, which is basically a lot of things that are not measured within the sort of the cell itself.
So I think we should leave some time for summary of comments that were made by people. I think we have here on the list some elements that are not necessarily already included in the things that are being looked at, and I think their role or their inclusion has to be discussed and addressed. And in a way, a summary is in this slide indicating that there's a lot of overlap and cross-reference between macroscopic and microscopic level, and this is all can be checked if we have the right technologies to do this, to look at the time resolution to the development throughout life, etc., etc. I'd like to stop here, and we'll have some comments about the points that were raised by the different participants in the discussion.
So one of the major points that we had a fiery discussion on was the nature of spatial versus genomic measurements. An anonymous person in the group described a bias in the HCA against spatial measurements, and I think the skin sort of highlights exactly how spatial, or an area where spatial measurements are incredibly important, potentially more important than the wider genomic assays. The example that came up that was really relevant was going back to some previously discussed work on the brain, where the mapping back to the Allen Institute spatial imaging was really the gold standard that allowed all of the genomic single-cell assays to be put into context. And so for the skin, for example, there is no current existing gold standard. So a potential proposal would be to go through and do imaging spatial measurements first and use that as a guide for future single-cell genomics. There was also a large discussion about the nature of the pilot draft phase versus the expanded phase. We could either start in the pilot phase by taking a few body sites and learning as much as we can about that, and then going wider in the full scale of the project, or in the pilot phase going for many body sites but only one or two donors and learning what the variability across body sites is, using that to guide how many body sites we need to assay in the wider project.
Okay. That's it, right?
Yeah, yep. That's it.
Are there questions, or we just go? If there are sort of quick questions, everything was so clear. Amazing. We are, if not, well, we're still here all day. So right.
Will do. Just one comment, because we've been through the perturbations, etc. There's one way to sort of embrace the perturbations of variability and to use it just in phase one as we want to see what is the range of effects that we can see, what is the range of possible variation, even though it's not at all powered to say this is what happens in different places, this is what happens as a function of exposure, et cetera, because those need much deeper analyses. So it's where having been gone through these discussions, it's important not to get fixated on power calculations to make sure that I know the influence of the microbiome in the first round, because that can sink the whole project, but simply to use this as a source of variability, which can then be explored in the second phase.
Which is true also for the geographical location, right? To a large extent, yeah. Right.
So did you guys consider a perturbation, a genetic perturbation like organoids, or matching an organoid to some skin and?
No. I think what went in the discussion was mostly the issues that are relevant for the intact skin in a healthy person. Now the skin, and for the skin, the perturbation is not something which is disease associated. I mean, this is the way of life of the skin. I mean, that's the physiology of the skin, and obviously the list could be 10 times larger than what's mentioned here. And I think the question of how do you stratify it in a program without exploding, the analysis is an issue. At what stage it should be done, or how do you connect it to some existing data concerning that accumulated over years about similarities and differences of different perturbations and so on and so forth. It should be reduced in an intelligent way, and we don't offer now how to do it exactly because that takes a lot deeper digging.
And can I ask another question?
Go on. I was going to add one point, but go on.
The microbiota, was that considered in this aspect? It does.
Yeah, it's
two.
one of the points that we discussed.
Sorry.
I think I was just going to add one more point about the organoids or skin equivalent, or however people call it within this field. And I think because the skin is extremely accessible and abundant, it actually lends itself very well to sort of having biopsies and also experimental model systems. I think skin equivalent or organoids are a great idea, but I think there's still a lot of challenges in terms of making it as similar to the kind of skin in vivo, because one of the issues is how do you reconstitute the immune cells within the sort of like, different layers of the skin.
Okay.
Oh, there's one more, Chris.
This.
There's one more.
I don't have a microphone, but I'll just yell. I think this discussion about spatial and tensile strength, it's very useful because it really, at least to my mind, and this is thinking a little bit coming from outside, it comes back to a point that Eric made last night, which is that we're really talking about a periodic table of what are the key atomic units, and the key atomic unit when we go from genes is what genes are expressed as RNA in a cell. And I think the minute we start layering all these other things on top of that, we start to move from the next level of periodic tables to how long is a piece of string.
Yeah, but you need the tools to make the difference between the residents and the visitors in terms of either molecular level or cells. And I think we should move on to a very different organ called the liver. Liver. And you invite later the kidney.
Okay. Okay, so the liver session was discussion was led by Ludovic Vallier, and Jay Shin took notes, and they can add points. I tried to capture most of what was discussed, but probably missed something. So liver is the major metabolic organ. There's something like 500 functions reported for it. It's also interesting as a regenerative organ. 80% of the liver can be removed and it grows back. We don't know how that works. It's quite amazing. There's a large and increasing disease burden. The global prevalence of non-alcoholic fatty liver disease is 25%, which is incredible globally. And then also liver fails late, and there's a lot of treatments. The major thing is you have to get a liver transplant. So the major discussion points were about getting liver tissue. What the first discussion point we covered was getting liver tissue. Liver transplants are a big activity, and so that's a major source of tissue. We can get healthy liver tissue or healthy, normal, fresh liver tissue from transplant programs. For instance, some surgeons can get an entire caudate lobe, so it's only one lobe of the liver, but it's a chunk of tissue. Whole organs are sometimes rejected for transplantation, and there could be different reasons for that. And so there's a little bit of variety in the health of those livers. The normal ones that get rejected are not healthy, and that's why they're rejected. There's a lot of biopsy samples and some healthy tissue from nearby cancer or non-cancerous tissue that's taken with a tumor. We discussed sample prep. This is a big challenge for liver because some liver cells are very sensitive to any kind of sample handling. Especially hepatocytes have low viability the longer you work with them. And so there's likely to be a big bias in single cell RNA-seq, at least, against specific types, and we don't know what that is. There was the discussion that maybe nuclei sequencing would help us understand that from nuclei sequencing from frozen samples. There's plenty of frozen samples available, but nobody's really done that yet, I think. So that is something that will be done. And then there's the idea that we, these biopsies and the caudate lobe is limited anatomically, so ideally we'd get a whole healthy liver for full detailed anatomical sampling. There might be special case access for healthy tissue. Some specific cases were mentioned where like a whole liver, a healthy liver is removed from a patient for specific medical reasons. There are some interesting, the next topic that we discussed is genomic and computational challenges. There are some interesting challenges that are particular to the liver. One is the sensitive cells that I mentioned, like hepatocytes quickly have low viability quickly, and we need to figure out how to handle that on the sample handling side, and also how to appropriately filter the data on the computational side to decide what cells we can keep and what cells we can't keep, because we get a large dispersion of or some variability in the health of the cells, and you want to maximize the number of cells that you keep. Binucleation and even like high ploidy numbers is common in the liver, so that might be an issue for nuclei sequencing as well. So cells have more than one nucleus. And there are spatial gradients that are important. So those have to be sampled and computationally assessed to understand differences between different zones. There's some interest scientifically in cell trajectory tracing, either during the regeneration process or during immune responses. Where do the immune cells come from? There was some discussion that maybe we need to include a challenge. So how do you do that? So do you try to initiate regeneration by some kind of injury, or that was discussed that might not be the best scenario because they'll get an immune response that might override any other signal you see. So that's a challenge that we have to think about both experimentally and computationally. We also had a question. People, I think, are getting ready to share data, not right away, but in the near future. And where do we put that data? Do we need a liver portal, or should we put data into the Broad single cell portal and just share it locally? and eventually the DCP will help with this. But, so there's a question, I guess, from at least this group of whether you're going to have tissue-specific portals or whether there's going to be a big center that you put everything. So how does someone who's a liver researcher come in and discover the information that they need? There's a wide range of interest areas, liver, of course, but also immune cells. Liver is a major immune organ. I think 10% of the liver is macrophages, and it's one of the biggest immune cell sources in the body. Stem cells, of course, developing liver. There's some interest from pediatric side as well. So those are different interest areas that could potentially form working groups in this project. We didn't discuss a lot about spatial sampling. I think there's a general sense that we need to do spatial sampling. We know that diseases affect different parts of the liver specifically, so presumably there's important differences between different parts of the liver that are important clinically as well. General sense is that we need to do that, but we didn't go into a lot of details, so that will come later, I guess. There's some need for, at least in my experience, sometimes we're talking to clinical collaborators and they don't necessarily get excited about this project, so we need to figure out how to excite them with applications and success stories from different organs. So can we talk about the blood system and leukemia? Can we talk about how understanding the immune system will improve rejection rates or reduce rejection rates in organ transplants? And we would love to hear, I think it would be great to hear from everybody in the community sort of these interesting clinical application areas, so that when we go and ask for tissue from a surgeon, they get excited about the project. Funding, currently liver is not a funded project through any specific HCA-related grant, so we need to think about how to move forward with that. Definitely everybody's interested in open access sharing, so those are 2 points that were briefly mentioned. We have a few action items. We want to create a central point for sharing experience, so there's a liver Slack channel, liver_pilot, so that's kind of got activated today. We want to share data, as I mentioned. We'll probably use the Broad single cell portal initially. And then we'd like to meet over lunch whoever's interested in liver to discuss next steps, because we want to move forward on this and figure out how to get resources together and work together on pushing this forward. Any questions or comments? Okay. Anything that I missed?
There's an anonymous commenter who would also say that spatial resolution might be even more important in the liver than anywhere else. There was a gorgeous talk from Shalaf at the single cell genomics meeting just before, and to me that really illustrated in what I would have considered to be fairly amorphous. Yeah, liver is a big hunk. There's incredible structure with incredible diversity of what otherwise would have been considered a single cell type, just from one end or the other. It may be one of the situations, because the cell types compared to other locations are probably not hugely numerous. So you guys could actually, one could suggest, could embrace from the start the spatial aspect, since you don't really have the neuronal diversity.
Yeah, so that's an excellent point. It was discussed. I should have shown a picture and to highlight, the known spatial gradients that exist. And so that's something I think we need to work on, because it hasn't come out as a major. People are not working on that right now, other than doing single cell RNA-seq and then going back and understanding mapping cell types to known spatial regions. But starting from a spatial sampling point of view, So far, I haven't heard of anyone actually doing that yet. It's an excellent point, though.
You asked a question about the Porta of our community, right? So I thought maybe Jeremy or Jonah or John would like to comment on that, on how they think about this. I will just say, because you also asked about the single cell portal, that we consider it in this context to be a temporary solution.
Right.
That as the DCP gets built, it will no longer be needed, but right now it's kind of convenient that there's something, so we keep maintaining it so that people have a place. But maybe Jeremy would want to say more from an architecture, whatever, perspective on what the thinking is like around the portals, because I think it provides a direct answer to this question. So you have to now go to the other side. That guy over there.
Yes. Thank you, Aviv.
Sorry.
Thanks, Aviv. No, this is great. Yeah, so we chatted about this during the session, but I'll sort of give the short version here, which is that I think this is the idea of a portal specific to an organ is a perfect example of what the portals in the DCP can be, the red boxes. And the idea is basically all the data across everything, all of the HCA will all come together and live across actually multiple clouds, but exactly replicated. That will be all the data for all of the HCA for all organs in the blue box, which is the primary storage system of the platform. But part of the design is that the data are on multiple clouds and to anyone widely accessible for the purpose of enabling lots of downstream use cases that include all kinds of sort of domain-specific or organ-specific or other kinds of particular ways people want to slice, analyze, visualize, or otherwise engage with the data. And I think a portal, as we talked about in the session, a portal specific to the liver, whether it was because of a particular way of slicing the data or a particular way of filtering on different QC metrics, or otherwise sort of portraying and visualizing and analyzing the data specific to this group, would be a perfect example of what the portals are for and how the whole platform is envisioned. So yeah, happy to chat. I believe there's a DCP like breakout roundtable thing during lunch, so this would be a great topic if you want to come to that and chat more about sort of what we imagine the sort of portal ecosystem to look like. But it's very much intended to be exactly for these kinds of use cases.
Okay, so we're moving on to kidney, I understand, next.
So I'm Anna Greca at Harvard Medical School in the Broad. Happy to be here. My partner in crime, we say, for the last couple of days is Nana Clatworthy from Cambridge. And we had a good discussion in the kidney cell atlas subgroup, which I wanted to summarize for you today. So just to kind of make a general point, because many of you may not think about the kidney very often, it makes you quite tired to think about the fact that the kidney filters 180 liters of fluid every day. So it has quite a bit of job to perform. There's about 500 million people around the world with kidney disease, and we haven't discovered any new treatments for kidney disease in the last 40 years. Hoping to change that. But you can see that where the need would be for us to really understand something about this organ in a more detailed manner. So the graphics for the white paper really were designed by Mena, who's really very good at visualizing things and designing figures, it seems. And so a lot of this is a credit to her, but we have envisioned two parts to the kidney human cell atlas. One will be the pilot phase, and the next would be the draft atlas phase. So I just wanted to describe the pilot phase, and apropos of that, just describe to you the different parts of the kidney that would be relevant to our discussion. And so, for the pilot phase, we want to really focus on two sources for kidneys. One would be nephrectomies, which happen because there's a tumor, but there's a lot of healthy kidney tissue next to that tumor, which we can take advantage of. The other is there are organ donors, there are kidney transplants that occur with quite a bit of frequency in most large centers. And many times, for various reasons, these organs are not utilized ultimately, technical reasons usually. So they are discarded, and we can take those kidneys as well and study them. And so the idea would be for us to really understand this complex architecture of the kidney. There are about 1 million such units in each of our kidneys called the nephron. So there are about 1 million nephrons in each kidney, and they're comprised of this complex structure. There's the filtration part of the kidney, which is called the glomerulus. So there's blood coming in, getting filtered through here, and whatever is discarded will be the primary urine that will go down this tubular structure. But whatever is clean, the clean blood will go out through the efferent arteriole, which is not really shown here, but this is the glomerulus, the filtering part. And then you have this long sort of tubular structure, which is defined already from old anatomists as having different parts to it, the proximal tubule, the distal tubule. The nephron resides in 2 segments of the kidney, which we call the cortex on the outside layer and the medulla. And ultimately, all of this filtrate that goes down this very, very intricate convoluted structure ends up in the renal pelvis and ultimately in the ureter, and then that will end up as the urine that comes out. So you can see that it would be a good idea for us to be able to sample all these different parts of the kidney. And we will come to this in a moment. And so for the pilot phase, we thought that we would try to get 30 humans, 30 kidneys that we could look at. And of course, our biggest goal is to really come up with SOPs that could be used across the board for this. And also to assess whether taking fresh tissue, which of course is optimal compared to freezing tissue and using it, how that would work. And so for the draft atlas then, we would actually move on to continue to have the same sources and process them in various ways, but in addition, take biopsy material. So a biopsy of the kidney is done by taking a long needle and taking a very small piece of kidney tissue as opposed to having a larger availability of tissue from an entire kidney coming out or a large discarded transplant kidney. And then the idea would be to fix samples from all of those. We'll talk about that in a moment for validation work further on. And so that would give us the opportunity to really, again, assess the same components of the kidney, but now perhaps doing it across more samples, sorting rare subsets. There are certain cells that are more poorly represented or more rare subsets of cells, so that we would need to look at those. Thinking about nuclear seq as a way to get past some of these problems with the representation, spatial transcriptomics that all the other groups have talked about, thinking a little bit about disease, as well as cases where there's immune processes in the kidney in the setting of transplantation, which may be of relevance to the field as another thought. And then obviously correlation with protein expression data. And then the spatial part is the microdissection of different parts of the kidney to look at that. So in the report back, I just wanted to discuss with you the points that came up in our group. So one of the things that Mena and I thought about and worry about is the issue of different ethnic groups being represented. We already know that there are certain, for example, risk variants in Africans of West African ancestry that predispose to kidney disease. That's a big one. There are probably many others. In the Asian population, IgA nephropathy is very common, so there may be some drivers of that disease as well. So we discussed the idea of needing to have a good representation of all different ethnic groups, and how do we do that? So at the moment we have Boston and Cambridge, but we would love to find a partner like the RICN. We already had some discussion about that in Japan, and maybe think about groups we could collaborate with for samples in Africa or Australia. And then, of course, the idea would be that through our pilot phase, we would be able to develop the SOPs that we can then share with colleagues across the world and be able to get samples shipped. That's where the frozen samples testing comes into play. We talked about anatomical mapping. Similar to GTEx, how can we make sure that this is captured when we collect the tissue? So there was an idea about an iPad using a 3D anatomical map of the kidney to then map that onto the tissue that is being collected, using a single page grid, which Kristen showed us earlier, to really determine the size of the kidney and the size of the extracted tissue in relation to that size. We talked about 4 sites that, based on the picture that I showed you, would make sense for us to collect tissue from. That would be the cortex, the medulla, the renal pelvis, where all the tubules collect, as well as the ureter, the ultimate structure that leads down to the bladder. We had a lot of discussion about how many cells and the depth of sequencing and how many cell types are there. It's not really known at the moment from old anatomy books and work that has happened. There was the idea that there are 26 cell types in the kidney. No one really knows. Mena has been doing some preliminary work, and our group as well, and there's a recent bioarchive paper that describes 16 cell types in a mouse kidney. But I think the verdict is still out on that. What is the rarest cell type? Is 1%? Is it 0.1%? What do we have to worry about in terms of being able to capture that? How many cells per type do we need? Kind of going into that calculation of how many cells are needed. It's not really known, and we'll have to keep doing our pilot experiments to figure that out. Integrating spatial transcriptomic techniques was something that we thought would be important. And also archiving SNAP frozen tissue from all the fresh tissue that we will be running in this pilot phase to start to do correlations. Also useful for validation studies. There's a huge abundance of FFPE kidney tissue out there in pathology labs across the world, but how to use that best. At the moment, we would favor frozen, especially if we're thinking about nuclear-seq. We don't know where the technology will go with FFPE, but certainly can be used for spatial studies and for validation work down the road. At least at the moment, that seems to be the consensus of how best to make use of that available tissue. We talked about sampling specific anatomical structures such as the glomeruli, the filtering units of the kidney. It's of special interest to me because I work on podocytes, which are perhaps the most rare cell population in the kidney, as far as anyone knows. So we will have to think about specific SOPs for the different anatomical structures. We talked about skydiving as a way to address the presence of rare subtypes that we would have to study and focus on. And then we went a little bit into what is normal, which was discussed by other groups as well. So we have defined our population as up to 55 years for a normal kidney right now, from what we will be shooting for. But what about older patients? There's this idea that, the aging kidney, there's some level of, disease that occurs as people age. But, could we go up to 60 or 70-year-old kidneys, and would that be informative in terms of the variation that occurs in what we would consider quote-unquote normal? Very prevalent conditions like hypertension and diabetes would affect the kidney. Is that something that is relevant to us? Would we do that in the draft phase, or would we wait to do that later? And having clinical data or phenotypic data on the patients whose kidneys we are analyzing will probably also be very important as we try to make correlations with the clinical picture. Similarly for transplant recipients or for other kidney diseases. So that was briefly discussed as something to think about for the future. We discussed, apropos of this, about metadata. I think that we discussed that this is probably going to be something that will have to be solved at the level of the entire HCA, but I think is very important. And how would we harmonize a system by which we can collect minimum amount of information, but also be able to perhaps go back and revisit phenotypic or clinical data for some of these patients. I think this is true for across the board, but certainly true for the kidney as well. Especially for diseases that are chronic, I think this will become important, because what we define as normal, when you go back into the clinical record, you may find out that this person actually had, for example, elevated blood pressures that were not known before or something like that, that would affect the outcome. And then in terms of analysis or analytical tools, there's clearly a need for generating a standard for that as well. For example, there's something very specific about anyone who has touched any single cell data from kidney, which is the fact that proximal tubules have an impressive amount of mitochondria. It's part of that really hard work of filtering that the kidney has to do. And so, how you interpret that in the setting of collecting data from kidney versus other tissues may be relevant to the analysis. I think that's where I will end. I wasn't sure if there were any questions based on this or points of discussion. Yeah.
So, for me, it's not a question, but a comment, both to you and to everyone else, regarding, both how many cells or all the issues of data design and standards. What the analysis working group would like from you and every other tissue atlas is less for you to sit down and try and figure out how many cells, but
Just do it.
no, not just that too. Start collecting.
Yeah.
It's going to be data driven, but more, what do you want to get out of it? So the analysis working group will put up the formal, as Barbara showed earlier, formal things to do it first, more simplistic and ever more sophisticated. But we need the guidance because we don't have the so we need to talk together.
Mm-hmm.
What percent cells? How rare do you want? Also, what are the important biological questions and things you want to gain? What would be successful atlas in terms of what you can learn from it, as well as all these points such as the mitochondria. So the more you let the analysis working group know your goals, your questions, your targets, your special things, we need you to figure out how do we sample this thing, because that's not expertise that we have. But if you could sort of distill your needs into more bigger picture goals and targets, then the more concrete how many cells and how the sampling need to happen to reach your targets is something that we will help you with. But we need your communication so we can do that.
Sounds good.
Just to comment on mitochondria, and this also goes PAN initiative. People have in the past offered me mitochondrial data because their single cell sequencing has failed and they just have mitochondria DNA or whatever, and they say, well, that's how we know that the analysis failed. I'm like, no, we want that. I work with Douglas Wallace. I don't know if a lot of people know him, but maybe he'll get the Nobel Prize someday, who knows. But we work for the Center for Epigenetic and Mitochondrial Medicine, and if it's known that a lot of pathologies, including probably in kidney, are linked to mitochondrial disorder. So it's something that we should keep in mind as we do these cell analyses. I know there's no particular metabolic arm right now, but I mean, I can volunteer the center, which he runs, and various people. I know there's Vamsi Mutha at Broad, and so if we could try to think about how to actually integrate mitochondria, maybe especially in your project, that would be really helpful.
That's a great point. Cool. Oh, sorry.
So I don't work on the kidney, but I know that the NIH has a kidney precision medicine initiative, and that they've been putting out RFAs, and I think have funded some U grants on a kidney cell atlas, and a lot of the discussions have been around many of these similar types of questions. So I'm just wondering how you integrate with them, and if there are lessons from the data sets they're already creating that could be useful for the pilots. Or
Yeah, it remains to be seen, but I think that, the more we put these ideas out there, I think the more likely, that we will be able to collaborate with colleagues who I'm very familiar with, who are spearheading some of those efforts. So I'm hoping for an all-inclusive approach. Any good ideas, wherever they come from, will be very welcome. Yes.
Okay, so my name is Herbert Schiller. I'm reporting on, okay, now we can see it. I have the pleasure to report on the lung breakout session. A bit closer, okay. And actually we had a fantastic talk by Jay today in the morning introducing the lung. And in our breakout session we touched on many points that were already mentioned. So we had planned to discuss all these things here. We couldn't really cover all of them. So the main points we came down to in terms of tissue procurement and data collection was the troubles in terms of extraction efficiency of certain cell types. So we also discussed pro and cons of nucleosec versus live. And then rather early on in the discussion we came to the point where we discussed whether we should have rather a few perfect healthy lungs screened in a very deep manner, so that we actually have the whole lung analyzed with imaging and sequencing concurrently. And the alternative to that is many lung biopsies, so we can have sections of different parts of the lungs from biopsies. And we also touched on that we all need to review our informed consent forms, whether some of the data release policies are really currently supported by what we have. And so maybe I go back a little bit to what Jay already touched on today in the morning. So the overall structure of the lung is, so we have this kind of space-filling fractal behavior of the airways that exist in 23 generations in the human lung. And it's really unknown what is the heterogeneity along the airway tree from proximal to distal. So this is certainly something we are interested in. And the real trouble here is that we don't know exactly how we can dissect the airways in the lower generation. So with bronchoscopy, we can reach down to the 5th or 6th generation of these airway branching events. And we could also get by surgery the more distal portions of the lung to look at the lung parenchyma. So the microstructure and organization of the parenchyma, the alveolar space, is of course also very complex. So we are also aware that we should really work on the spatial transcriptomics methods in terms to define these micro niches that may exist. And then one important point in the discussion was if we go for this kind of dual strategy, that we try to have one or two lungs really deeply and very well characterized, we could have the other arm which relies on the surgeons that give us tissue. And the surgeon can tell us from the anatomy book this bronchopulmonary segments. So we actually, by retrieving tissue from the clinics over time, could screen over the whole basically organ. And we also get more information about inter-individual variation if we go along this strategy. So in that case, we could retrieve healthy tissue from pneumothorax patients or uninvolved tissue from cancer resections, where actually we also had a discussion how well we can accept that this tissue is truly healthy. And that brings also the point that we need small disease cohorts to define the extremes of the disease condition. And we could learn from the data whether the supposedly healthy tissue that we obtained in the uninvolved area from the tumor resection might have been a real healthy tissue. So that comes down to these different strategies of tissue procurement. One way for this very deep sequencing is of course the diseased organ donor. And then sometimes we can get tissue from transplant lungs where the lung is actually too big for the donor, for the recipient, and it's trimmed a little bit. So sometimes we can get tissues from here. Then there is direct sampling of airway wall tissue, so bronchoscopy, which can give us also rather healthy tissue. And as I already mentioned, the parenchymal tissue we get from pneumothorax or uninvolved resection material. So we tend to think that this dual approach could be a good way. At least that was my feeling how the discussion went. And it seems to be also important to have cryopreservation and FFPE blocks of all the tissues that we are going through. So currently in Munich we are doing fresh lung tissue, but we also have this bioarchive effort that we could go back to the same tissue for some imaging approaches. I tried to find out from old literature how many cell types were actually described in the lung, and usually you read about this magic number of around 40 cell types. And already today in the morning we heard by sequencing a 10,000 airway wall single cells, it was possible to already detect new cell types here. So we think the number of 40 will certainly increase. And another important parameter for the skydive would be the relative frequencies of the cells. So you can find in some old lung stereology literature and electron microscopy study the estimated relative amounts of the main cell types. And I just had a look in the first data sets we get from human lung, whether actually the relative proportions are in concordance with what we would predict. And that seems to work quite nicely. So the two main epithelial cell types in the lung parenchyma, the type 2 and type 1 pneumocytes, have approximately a ratio 1:2 from the classical studies, and we see exactly the same ratio in the dropsic data, and then also the ratio of the airway basal cell to the type 2 cell seems to be quite correct. And this little tool was already mentioned today. If we think about the possible rarity of different cell types, so we know about some stem cells that might be 1 in 10,000. So in that case, with this model, we would need to screen 250,000 cells to see 10 cells of that rarity. So what we think we have to do is in an initial phase do the non-stratified sampling, but most certainly we have to go to some kind of stratified sampling strategy. And the two ways we think about it is, as I already introduced, the spatial stratification along the airway tree, and then the lineage stratification with the classical main cell lineages. I have one slide on a new spatial technology. So we have in the lung working group, we have now Tusha Desai from Stanford, and they developed, so this is unpublished work, and they developed a new technique for the in situ hybridization. I just wanted to share this with you. I'm not an expert on this technique, but basically that is using this proximity ligation strategy that was also used for antibody colocalization. And this rolling PCR amplification can actually increase the signal to noise ratio quite a bit. So he's getting fantastic data from fixed lung tissues, where he can actually, with in situ hybridization of these markers here, can nicely distinguish the different cell types and also identify really rare cell types. So this yellow one here is a bronchoalveolar stem cell in the bronchoalveolar duct junction, and that has been described exactly in this position based on protein methods. And so they could screen thousands of cells here with their multiplexed new in situ hybridization method. So what we plan to do is also apply that for the lung, obviously. And then the last point we touched upon is how much of disease do we actually have to sample in order to understand what represents the truly healthy condition. So this slide is just to summarize that chronic lung disease is one of the major health burdens. And 4 million people die prematurely every year from chronic respiratory disease. Main killers here is definitely COPD, but so many people suffer from asthma, and lung cancer is the most deadly cancer we have. And so that was just one topic in the discussion, if we should include small cohorts of COPD and other type of main lung diseases. So for instance, Martin Nabein in the Netherlands, they are already having airway wall biopsy data from both healthy patients and asthma cohorts. So actually this data could be used to understand a little bit better what the asthma condition could look like. And in terms of disease modeling, I think it's very important to understand the difference between human and mouse lungs. So we actually started correlating some of the cell types we observe in human lungs and mouse lungs, and just look for species-conserved core signature genes that we can use for the spatial methods. So finally, somehow the action items we have on the short term is increase the communication between the different groups. So it was quite a big group in the lung breakout session. And we also have that Slack channel, and basically would want to start sharing the data we already have. And for a first phase, one possible strategy here is to sample something like 5 sites along the airway tree, so the tracheal rings and primary bronchi, the 3rd and 6th generation of the bronchi, dissected small airways, and then the lung parenchyma as a whole. And as I already said, in a second wave, we could have a more stratified approach, and then start also to work on the spatial methods. I think that's almost the final slide. So what we want to have is now a consensus for tissue sampling across the different centers. So we definitely observe problems with live tissue extraction, that we miss certain cell types with some of the protocols. And we also want to establish consensus lineage signatures for the spatial methods. And then it was not really discussed how long we would need for a first phase. So if we would aim for 1 million cells, if we can do that already in 2018 or later. So that was not really discussed. I just had it on the slide. So I think with this I'm happy to take questions. Thank you.
So I might have missed this in all of the presentations, but is there an implicit assumption that the genome is going to be sequenced of all of the individuals that consent to be part of this project as well? Or is that something that we should be considering when we think about what information is going to be allowable? Because it would seem like a little bit of a missed opportunity if we don't have that information alongside the single-cell RNA sequencing where possible.
I'm not sure if I'm the person who should answer that. But
well, but you can answer it in terms of the data that you might envisage collecting. Would you also envisage
so
doing genome sequencing as well?
We are currently not doing this, so we do the. I'm not aware. I'm speaking for the whole group, so I don't really know if anybody is doing that. So we are not doing that currently.
I mean, maybe I can ask Aviv or Sarah then. I mean, would you envisage that the individuals that are part of this project should also have their DNA, the genome, available as well for analyses?
The present session comes out perfect. Thanks a lot. So there's one person out there who's paying attention. No. 15. Okay. Cool. No, it's lots of people also watch later and so on. So I think it's great to have this streaming set up, because we've had hundreds of sort of compliments on it from Stockholm. So we should continue this tradition of openness with streaming. John's question about DNA sequencing, I think, is great. It's really, really interesting, and there are many levels at which that could be done, ranging from genotyping to actually, deep exome sequencing, or deep whole genome sequencing of individual biopsy samples, and then there's the question of, can you then map somatic mutations basically back to the single-cell RNA sequencing and track clones and so on, and potentially the cost isn't so huge that's prohibitive, and that's a strategy that I think we need to think through as a community, at what level we want to have that complementary data along with the samples. And for clonal samples, it may make sense to even sequence many individual biopsies from a given donor. For clonal tissues, for other tissues, it may be less interesting, but certainly a general a general strategy for the genetic side of things is worth thinking about now already. And this could be the remit of the analysis working group, Dana and John, or it could be actually a separate working group that's set up to think this through, the human genetic side of things from the start. So I think that's something that's worth, John, over to you again, really.
Quick short question, because we have 1 hour of lunch now. And we have to get back at 2:30.
Okay, it's very short. The whole big question is to be quick, real follow-up for people who are interested. So one of the cohorts that the pediatric group can offer is whole genome trio sequencing of both parents, child, and multiple tissue somatic sequencing, as well as single cell analysis. We're going to try to do this, and if anyone's interested in this, it's a disease called DIPG, which is a fatal tumor type in children. We're going to have a good-sized cohort of rare DIPG samples. So if you want to work out a protocol, if you want to, so if you want to work on protocols for this, maybe we could talk, because we already have a grant going called the Gabriela Miller Kids First Data Resource that was recently awarded, and we're the Data Resource Center, and we also have additional money that BGI was very kind to donate for additional sequencing.
All right, so let's thank everyone.